Okay, so let's start the genetics PYQ solve, and this is from DBT BET question paper. So we will solve DBT BET genetics question paper questions from PYQ, and this is actually probably 2008 to 2022. So let's start with the first question. Galactosemia is a recessive human disease that is treatable by restricting lactose and glucose in the diet. A couple is heterozygous for the galactosemia. Okay, so the couple is, let's suppose, capital A small a and crossing with another heterozygous, that is capital A small a. If the couple has 4 children, what is the probability that none of 4 will have the galactosemia? So the offspring from this uh, couple can be galactosemia positive or negative, and galactosemia negative, the chances of galactosemia negative is 3 by 4, because capital A, one genotype is capital A, that will be is galactosemia negative, and another capital A small a, that is the proportion of 1 by 2, that is 2 by 4. So the total is 3 by 4, and there are 4 children, so 3 by 4 into second child is also 3 by 4, into third child is also 3 by 4, and fourth child is also 3 by 4. So the probability of being children consecutively galactosemia negative is 81 divided by 256. So option 4 is the correct here. Second question is saying that what is the number of second question is saying that what is the number of phenotype in F2 of dihybrid will be. So this is very basic question. That's number of phenotype in F2 in dihybrid. If we do a dihybrid cross, then what, how many types of phenotype we can get? So that is 4 types. The resultants are 9 is to 3 is to 3 is to 1. So one phenotype is 9, one phenotype having 9 types of genotype, one phenotype have 3 types of genotype, another phenotype have 3 types of genotype, and another phenotype have 1 type of genotype. So 4 types of genotype phenotype can be present. What is the incomplete dominance? Is the What is the incomplete dominance of monohybrid? The number of phenotype in F2. In incomplete dominance, we can see that dominant character, that is, one is maybe red, another is maybe white, and another is pink. The in between character of two allele, two trait. So we can get 3 types of phenotype: red, pink, and white. So what is the 3 options? Option 3. Mendel's law of segregation is applied to behavior of chromosomes during cell division. Means that Mendel's law of segregation, that is Mendel's first law, allele of gene separates from each other when homozygous chromosome separates in meiosis. But allele could be separated when the allele when the actually alleles are separated. I'm talking about that if crossing overs are there. If there are crossing over, then the homozygous chromosome, then one chromosome can have 2 type of allele, and then this option is absolutely incorrect. Alleles of genes separates from each other when chromatids are separated, but if crossing over is not there, then this is not true. Alleles of genes separates each other when homologs separates in meiosis 1 or when chromatid separates in meiosis 2 if there is crossing over. That is what we are talking about. So option 3 is absolutely correct. Option 3 is absolutely correct. And alleles of gene separate each other when chromatids separate in meiosis 1. But in meiosis 1, there is no chromatid separation we can found. In meiosis 1, there is no chromatid separation. So this is again false. Gene related to through descents from a common ancestral gene. Gene related through descent from a common ancestral gene is we call what we call this is orthologous. This is orthologous. If we see a clade having one ancestral gene into the new species, then that is called as orthologous. And for this concept, you can check this image. See, homologs when can be further classified as homologs can be classified as three types: orthologs, paralogs, and analogs. So, if homologs, what is one? What is homologs? What is homolog? Homolog is the gene or proteins that look or act similar because they are evolutionary same origin. Now, what is orthologs? Orthologs is speciation after evolution. So, there is a common ancestor, and from this two genes are speciated. What is paralog? After gene duplication, if there is a gene duplication occurs and one is separated into another and another is separated into another. So one person can have this both the genes. And what is analog? Analog is two different ancestral genes can act as similar. Can act as similar. That is analog. Now let's go to the question again. Next question, question number 6. An individual has the genotype capital A, small a, capital B, small b, small c, small d, capital E, small e. Assuming the independent assignment of what the frequency of gamete have will have the genotype small a, small b, small c, small d, small e. So from this genotype, we have to prepare all small gamete. So the chances of coming small a from capital A small a is half. From chances of coming small b from capital B small b is half. From the what's the chances of coming small c small d from small c and small d is 1 and 1, and chances of coming small e from capital A small e is again half. So what's the resultant is 1 by 8, and here's the answer, option 2. Next, the athemic nude NU mice is difficult to breed and maintaining because the homozygous NU females are sterile. So homozygous NU males are not sterile. How will you propagate it to get maximum number of NU mice progeny? So to get maximum number of NU progeny, we can use NU male and heterozygous female. So let's see the answer. By mating normal male. No, we are not. We will not use normal male. By mating homozygous NU male to heterozygous female. We already talked about that, and that's the true. Mating heterozygous male. Why will use heterozygous male when we can use homozygous male? Because the question is saying homozygous females are only sterile. And next, mating heterozygous NU male, no. Next, what, which one of combination given below represent population with similar genetic composition? They are having similar genetic composition, and I also don't know all of this uh, abbreviation. So this question is not a very appropriate question because this abbreviation should be mentioned. But as I have researched about this question, this DH is double haploid, double haploid, and there is no chance of having similar genetic composition in the F2, that is filial 2. So the only chance is there in option 3. I can't explain this question very in a good manner. If you can comment, because this question is not very classic question or very good question. So let's go to the next question. Given that husband and wife both heterozygous for a recessive allele albinism. Okay, so both are recessive. So both are heterozygous. Sorry, both are heterozygous. That means again capital A small a cross capital A small a. If they have dizygotic twins, what is the probability that both twins will have the same gametophyte for pigmentation? So if this both should have to be the same gametophyte, same genotype, same phenotype. Sorry, same genotype, not same phenotype. So if we are seeing that childs are normal, the childs are normal. So normal child, probability of normal child is 3/4, and again probability of normal child is 3/4 because two childs are there, twins. They are saying dizygotic twins, so two different childs are there. That is what, 9/16. And if disease child, then 1/4 and 1/4 because both have to be same. So that is the probability is 1/16. And they are saying about, they didn't said about any which pigmentation should be. The child could be normal, the child could be diseased. So if child, so the sentence is the having, we are getting, that is the child could be normal or diseased. So in that case we have to add these ratios, 9/16 plus 1/16, that is 10/16 and 5/8. Is there in 5/8 options? Yeah, option A. Option A, 5/8. Right. If there is any problem to consider to understand this question, this answer, you can comment below, and I will try to make a separate video for this only question. Yeah. Let's see the next question. Consider three independently assorted gene pairs, capital A small a, capital B small b, and small c, capital B small c. So the probability of obtaining the offspring, capital A, capital S, capital B small b, and capital C small c, from parents that are capital small, capital, and capital small, capital small. So if we cross only capital A small a and crossing with capital A small a, that is the genotype of parents, capital A small a and capital A. So what is the ratio of coming capital A into the child, that is 1 by 2, right? Now if if we cross capital B small b, cross capital B small c small b, and we have to get capital B small b into the child, so that is the probability of 2 by 4, that is 1 by 2 again. And we are crossing capital C with capital C small c, so the probability of having capital C small c is 1 by 2 again. So if we cross all of these, 1 by 2 into 1 by 2 into 1 by 2, we will get 1 by 8, and that is the probability. Option 3 is correct. Assume gene A is dominant over capital A, okay, and gene B1 is co-dominant over capital B. In Pretonia, we are crossing capital A, capital B, B1, B2, cross small a, B1, B2. So you can see that capital A are homozygous, and capital small a are homozygous. So from this cross only, we can get all capital A, small a progeny, and capital B, small b. No, sorry, B1, B2, and B1, B2 are heterozygous. So these are, as these are codominant, we can get we can get three types of progenies. That is B1, B2, that is B1, B1, and another is B2, B2. And this is codominant, so we can get three types of progenies, and that is 1 is to 2 is to 1. Option 3. That is the ratio of codominance, right? Next, assume that a marker M1 is present 5 centimeter away one side, okay, so that is 5 centimeter away from one side of gene X. So let's assume that gene X is here, here is M1, and this is 5 centimorgan, sorry, not centimorgan, centimeter, that is centimorgan, and another gene is there, that is M2, and this is 10 centimeter away on the other side. Yeah, we have also drawn this 10 centimeter. Now the donor genotype is M1 M1, XX, M2 M2, while the recipient has small m1, small x, small m2, small m2 genotype. A cross made between two two of these individual, the F1 crossed is crossed to recipient. The progeny of the crosses between 100,000 plants, how many plant will progeny have M1 M2 while the desired gene is absent? Okay, so they are talking about that we have one donor and one recipient, then we have crossed, and we got F1 progeny. Now we are trying to cross F1 progeny, we are trying to selfing with the F1 progenies and producing 1,000 plants, and how many of these plants will have the markers but not but absent the desired genes? So only this could be happen if double crossover is present. So here is one crossover and here is one crossover. Only then the things is happening when markers are present, but genes are not present. Genes is not present. So the chances, the percentage of chances of having these two these two uh, uh, crossing over is 1 by 5 into 1 by 10 into 100. That is what 100 not, there is thousand plants, sorry, the total plants is 1000. So we can uh, divide that, we can divide 1000 with 50 and we will get 20. So 20 plants are double crossover, and if we see double crossover, the double crossover plant will be M1 small x M2, as well as the double crossover plant will be small m1 capital X and small m2, and they are asking about this plant only, that is having the marker but not present the gene. So this is the half of total double crossing over. So half of 20, that is 10, and this is also half of 20, that is 10. So what is the number of our desired plant? That is 10. Option C. Again, we can solve it through steps and very precisely. So if you couldn't understand, you can comment below and we'll make a separate video elaborating all the steps one by one. Let's go over the next question. Parent who appear normal have a child with sickle cell anemia, which is an autosomal recessive trait. The woman is pregnant again and told that she is carrying fraternal twins. Fraternal twins is dizygotic twins. What is the probability that both twins will develop sickle cell anemia? So, as we know that parents are normal, but they are having a child with homozygous recessive parent, homozygous recessive character. So, both are obviously capital small a, both are obviously heterozygous. And again, if they are heterozygous, what is the chances of both the twins is diseased? The chances is 1 by 4 into 1 by 4, that is 1 by 16. Option A is the correct answer, right? The deletion of gene X in mice did not result any abnormality. Further, to investigate the function of gene, at least 5 null mutations of the same sex are needed. What is the minimum number of pups from heterozygous parent will be screened to obtain the required number? So, to get the maximum progeny, we will cross with capital A small a and capital A small a. We have to cross with heterozygous to get small a, and we will get 1 by 4 small a progeny, and to get at least 5 null mice, we have to cross, we have to get total 20 progeny, right? So if we see total 20 pups, there we can find 5 null mutation mice. But the question is saying, that the at least 5 null mutation mice of same sex. But if we see total 5 mice, can we get all with same sex? No. So to get 5 mice from same sex, we have to increase the total number of mice. So let's see about the if you see, there is 20 and next is 40. So 40 could be the answer, because 20 and 10 could not be the answer, and 80 is very high. Next, what will be the probability of obtaining a plant with capital A small a, capital B, capital C, small c genotypes of trihybrid from this parent? So there is we have this parent and we have to get this offspring. So from capital A small a into capital A small a, as both the parents are same, we have to get capital A small a, and the probability of getting capital A small a is 2 by 4, that is 1 by 2. Okay, capital B small b into capital B small b, and we have to get capital B, and this is capital B, and this is the chances of 1 by 4. And from capital B capital C small c, and cross with capital C small c, and we have to get capital C small c. The chances is 1 by 2, that is 4 by 2. So the total chances is 1 by 2 into 1 by 2 into 1 by 4, right? So that is 1 by 16. Is there an option? 1 by 16? No, this is 1 by 64, 0 by 64, 8 by 64, and 4 by 64. So yeah, this is 4 by 64 means 1 by 16, and 8 by 64 means 1 by 4. So this is not the answer. Option A is the answer, 1 by 16. Next, in rice, while pyramiding 3 gene of trait, 2 donors were used. One donor carries 2 desirable genes, that is on chromosome number 2 and chromosome number 4, while other donor has 1 desirable gene on chromosome number 3. Both the donors were crossed to produce biparental F2 population. What is the theoretical expectation of individual carrying all the desirable allele homozygous condition on one of them, or one out of? So there is three point crossing over, 3 point hybridization, 3 point hybridization, and they are asking what is the probable percentage, what is the probability of having all dominant character in one plant. So if we see the all dominant character in one plant in 3 point hybridization, the probability is 1 by 64, as we all know. So option 3 is answer. We can describe it in elaboratedly. So you can comment. If the allele A is incompletely dominant over capital A, what is the expected in progeny of two heterozygous parents? Same phenotype again heterozygous. Oh, that's exactly correct. In completely dominant character, we can see 1:2:1 ratio for genotype and phenotype both. So this is absolutely correct. 2:1 phenotype ratio no, 3:1 phenotype ratio no, 2:1 homozygous dominant and intermediate phenotype no. Let's see the next question. Allele A dominant over allele small a and results dark skin pigment. So allele A, capital A, is producing dark. In the mating of capital A small a with capital A small a, if 6 offspring are produced, the probability of having dark pigment is probability of all having dark pigment. So all, if we get all the dark pigment, so if there is capital A small a cross capital A small a, the probability of dark phenotype is 3 by 4, and the probability of all 6 dark is 3 by 4 into 3 by 4 into 3 by 4 into 3 by 4 into 3 by 4, that is 3 by 4 to the power 6. And if we calculate this, if we calculate this will come around, come around 0.177, and that is very near to 0.18. Option 1 is correct. Next question: which one of the following statement is about alleles are not true? Which one of the following statement about alleles are not true? They may occupy different loci in the same chromosome. How? How one allele or two allele can occupy different loci of same chromosome? This is absolutely incorrect, and we don't have to even read the other statements. But still, they may show codominance. That's true. They may be dominant over another. That's true. They may be several at one locus. That's also true. That is multiple allelism. ABA blood group. Next, a combination of genes, combination of gametes that can be formed by the genotype capital A, small a, capital B, small b, capital C, small c, capital D, small d, capital E, small e, capital F, small f, capital G, small g. That's actually very true. Very easy. 1, 2, 3, 4, 5, 6, 7. Total 7 heterozygous are there, and we have a formula for this question, that is 2 to the power n. N is number of heterozygous. If you want to check how many types of gamete could be produced from a genotype, you can use this formula, that is 2 to the power n, and n is number of heterozygous. So here is 7 heterozygous is there. So 7, 2 to the power 7, 2 to the power 7 means 128. 2 to the power 7 means 128. So 128 types of gamete could be produced here. Genes related through vertical descendant. Genes related through vertical descendant from a common ancestral gene. We have already seen this concept, and genes are related with vertical descendant was what? Orthologous. We have already did this. Next, genes are related through gene duplication. Whenever you get the term gene duplication, you can do. Next, a geneticist interested in immune function induced random mutation in number of specific genes in mice and determines which one is resulting mutant mice have impaired immune function. This approach is example of reverse genetics. This approach is known as reverse genetics. That is when we mutate one gene and see what is not happening there. Right. Next, breeding of capital A, small a, and capital A resulted 5 offspring. What is the probability that exactly 4 offspring will have capital A, capital A? So for this we have to use binomial uh, equation. So binomial equation is, there is 5 total offspring, so 5 factorial divided by 4 factorial of capital A and 1 factorial of small a, crossing the, what is the ratio of getting capital A, small s, getting capital A from capital A, small a, and capital A parent, that is 1 by 2 to the power 4, cross again 1 by 2 for the another genotype, and that is to the power 1. So if we write this 5 factorial into 5 into 4 factorial, we can do 4 factorial into 1 factorial, that is 1 into 1 by 2 to the power 5. We can write this like that, and 4 factorial and 4 factorial we are cancelling out. So 5 into 1 factorial into 5 means what? 32. So this is 5 by 32, and option 4 is the correct answer. That's how you can solve all the questions from genetics, Mendelian genetics mainly, and we have discussed all the Mendelian genetics question here. So let's meet in another video with the next chapter, multiple allele and gene interaction. For now, thank you so much and share with your friends. Thank you.